Ladies and gentlemen, welcome to the first talk of this session. The speaker is Professor Alexandre Douplik from Toronto Metropolitan University. He is an expert in biophotonics and is going to talk on experimental models and applications in biophotonics for the next 45 minutes. Please join me in welcoming Professor Alexandre Douplik.
Thank you. Thank you very much. And I'm enjoying coming here and making the presentation. And today I would like to talk about experimental models and applications in biophotonics. And I would like to tell you about the project which started a few years ago. And it started basically as an idea which was first analyzed and then simulated as a pure kind of like virtual realization or utilization of this idea. And the mathematical simulation through approximate analytical solutions and then through numerical simulation has shown that this idea might work. And then we proceeded with experimental work, and then we started to use a device, a prototype, what we created for some biological and biomedical applications. And today it's going to be a story about one idea which has been proved first mathematically through numerical simulations, and then it was implemented as a prototype device and collecting data and analyzing this data. So let's begin with our journey. Today, the technology what I'm talking about is called ballistic photon imaging. I will in details explain what is that in light scattering media and biotissues. Okay. So on the first slide, also you can see the place where our university is located. I'm representing Toronto Metropolitan University. This is the second university in downtown Toronto. We have roughly more than 50,000 students, and Toronto is a capital of Ontario province in Canada. All right, so this is, by the way, our Student Learning Center, erected maybe just a few years ago. And let's talk about what is the main challenge in biomedical optics. We are dealing with light, and light that interacts with the biological tissue. And the main challenge is that light penetrates into the tissue at a very small depth. Generally speaking, it's only a few millimeters max. Because of few reasons. So reason number one, there are a lot of while light penetration into biological tissue actually reaches such a small depth because of two things: light scattering and light absorption. And the next difficulty is that there is no dominating contributor into this limitation. Both light scattering and light absorption are almost equally important and comparable contributors in the restriction of the penetration of the light inside the body. So, how light scattering and light absorption affect bioimaging? That's basically what we are going to concentrate today, and the application of bioimaging. So light absorption is usually a localized and contrast-based phenomenon that gives, for instance, an opportunity to image blood vessels, nerves, extracellular matrix, bones, actually, in our body, there are a lot of structures that absorb light in visible range. And visible range, it's approximately between 400 and 700 nanometers. So that's quite a narrow. And the second component is light scattering. And light scattering, it's not localized as absorption and not contrasting, but the other way around. It's a diffuse based. So due to the difference in refraction indices between cell in and cell out, intracell organelles, cell and organelle membranes, it actually disguises and makes everything blurred in a way if we would like, for instance, to provide an imaging based on the absorption structures, which are, once again, are localized. All right, so the bottom line, what we can say so far is the bioimaging contrast due to the light absorption is reduced by light scattering. So just to illustrate this idea, we can compare a human skin. So on the left, right here, that's how we see our skin with an unarmed eye, right? So we don't see anything because the skin and the surface of the skin, and more exactly epidermis, has a very high scattering. What if we had not the scattering? In this case, we would see all the blood vessels in the skin quite clearly. And, well, the picture was generated by AI, and that's kind of a simplification, of course. But just to give you an idea, with our scattering, that's our surface would look like. Yeah, very nice, a good contrast, so we could easily see what's wrong with our blood vessels, but instead we see that, and that is because of the light scattering. So if we would like to provide the imaging, definitely we should reduce somehow the scattering, and that's what's called clearing or optical clearing. So it meaning that somehow we reduce the scattering in order to reveal the absorption contrast. So what we can do in order to reduce the light scattering? So first of all, we can apply optical clearance and reduce the refraction index difference. In this case, there will be no physical basis for the scattering, or at least we can reduce it. So the second approach is, for instance, applying a needle endoscopy. So we can penetrate inside the skin and see actually some deep structures because we approach that with a needle. So the other approach is time domain gating. Less scattered photons actually arrives faster. If we make gating, for instance, the fastest photons, in this case, we obtain less scattered photons. Less scattered photons would provide us with imaging that is basically based on the contrast of the absorption structures. And in this case, the detector collects those photons and facilitates basically the imaging. Some approaches like Anderson localization and so forth, I'm not going to touch base with that today, but those are basically computational approaches of a different kind. And those are also exist when, let's say, an imaging, when scattered and non-scattered light is collected, and then due to this processing, or at least we can retrieve to try certain unscattered photons. So the other approach is what's conditionally at the moment called in our group as a ballistic photon imaging. So that's exactly what I'm going to focus today, and this is gating less scattered photons via restriction of the detector's numerical aperture. We will be talking about that in details, and fortunately today I have like almost 45 or probably slightly more minutes, and I will explain exactly what the idea of ballistic photon imaging. All right, so first of all, let's figure out why we have image blurring. We have it because, for instance, like in general, this is not specifically for biological imaging or whatever, but in general, if we use a camera, and now we have all digital cameras, of course, so they have pixels. Each pixel also has a numerical aperture. If those numerical apertures overlap, so as a result, we have blurring. So what we can do with that? Before we proceed to the idea of ballistic photon imaging, I would like to explain certain terminology and maybe a certain theory behind that. So then we can run kind of like very confidently because we would understand what's behind. So first of all, coming back to the absorption on a fundamental point of view. So let's say electromagnetic radiation absorption on a particle would be characterized by the certain cross-section. So this is basically a probability of a photon to be absorbed by a certain object. And the overall absorption cross-section, this is a product of absorption efficiency or probability actually to be absorbed and geometrical cross-section. If we multiply the number of particles, each of them has a certain absorption cross-section, times the number of particles, we obtain a parameter called absorption coefficient. And the absorption coefficient characterizes, and this is an intrinsic property that does not depend on how you measure that, but what's more important about absorption coefficient, that's absorption mean-free path. So this is the mean distance traveled by a photon before absorption. And that's exactly the inverse value of the absorption coefficient. And in physics, we apply the absorption description of the process through the differential equation of the first order called Birnbaum law, and that is a representation of that. So the solution of those, of course, it's exponential, where the absorption coefficient, it's a negative exponential, and it's a product of absorption coefficient times the path length of the light. And this is just, I hope that it will work. Yeah. So this is just an illustration how the absorption actually occurs. So it comes inside the absorbing media, and then, as you can see, this is a negative exponent. This is a probabilistic process, and once again, it's characterized by the absorption coefficient, which is measured as inverse millimeters or inverse centimeters. Okay. So the measurable parameters that are based on Beer-Lambert law are usually transmittance and absorbance. That basically characterizes exactly the losses of light when light goes through a biological object. And this is an example of absorbance of probably one of the most important absorbers in human body. Those are hemoglobin. This is a part of erythrocytes, which provides basically the oxygen supply and the energy for our body. And those are two forms of hemoglobin, oxygenated and deoxygenated. So the absorbance makes sense for a single molecule times the concentration of this molecule, either molar or number of molecules. And this is kind of like on a wider scale of wavelengths. You can see in the visible range between 400 and 700. So that's what I just was showing you, the hemoglobin. And those are mostly representation of water. And overtones of oscillation of water molecule, they have the highest peak of absorption as approximately 3 micron, which is near infrared and not a visible range. Okay, so that's about absorption. How about scattering? Light scattering, based on the Jablonski diagram, is basically the process when a photon is absorbed and then re-emitted without the loss of energy. So this is shown here, the outmost left transition, and that what differs from the non-elastic, so that's called elastic scattering, differs from non-elastic scattering when it's excited to the next level, coming back, but there is a certain loss of energy. And that's, we are talking about Raman scattering and infrared spectroscopy. Okay, so in general, the base for the light scattering can be described polarizability of the molecule. Yeah? So the higher the polarizability, the higher probability to be scattered. And that's what we calculated working on the project, the angular dependence of scattered light. Basically from dipole, and this is here, and quadrupole, octopole, and hexen decapole. And that is scattering profiles, normalized intensity over the solid angle of various multipoles. So from dipole to quadrupole, again octopole, and sum of few multiples. So the point is that depending on how many dipoles we consider in the system, so generally speaking, the scattering phase function, as you can see, is pretty different. And the scattering cross-section overall is described in a similar way as the absorption. There is a scattering cross-section, which is, as you can see, the ratio of the scattered power over the initial power. And that's basically using the same diagram as was for the absorption. How we can take that into account, that's the next question. Well, we apply again the same approach. Once we know the scattering cross-section for each object, and each object can be characterized by the ensemble of dipoles, and knowing the number of the object. So as a result, we can multiply the scattering cross-section of a single object times the number of the object, the scattering objects, and we obtain a scattering coefficient. And the physical meaning of the scattering coefficient is the same as for the absorption, but just for the scattering. So it is going to be an average distance between the scattering events. And what is shown here is basically a penetration of light of a different color inside the human skin. Okay, so now let's fuse them together. Because in biological tissue, what I mentioned before, both absorption and scattering processes occur, and those are comparable. So when we superpose both the absorption and scattering coefficients, that's called attenuation coefficient, and that's actually what we can use to a certain extent. as a Beer-Lambert form when instead of absorption coefficient only, we use a superposition, the sum of absorption and scattering coefficient, called attenuation coefficient. And the important parameter is what's called a transport mean free path. And transport mean free path, so there are like two parameters to characterize the scattering properties of biological tissue or any type of object. It's a scattering mean free path, which is again the distance between two scattering events, mean distance, and the transport mean free path. As you can see, instead of like mu s, which is a scattering coefficient, it contains mu s times 1 minus g, and g is a mean cosine, so-called mean cosine of the scattering angles, and also called anisotropy coefficient. The point of transport mean free path is that's the distance when the photon entering the scattering media becomes totally randomized and the original direction actually lost. So it becomes totally diffuse. So meaning that at that moment, basically we lose the contrast at all. So while here in the beginning we still can provide a certain crosstalk because the fraction of low order scattered photons are presented here. After transport mean free path, it becomes totally diffused, and the recovery of the original contrast is practically impossible. And the scattering factor and anisotropy factor, just to explain a little bit what the G is. So this is a scattering angle. Remember, for the dipole, we were building a diagram of scattering angles. So the average value of this angle for the ensemble, that's called for the object, and basically this is the parameter for a single object, is provided by the phase function. And the phase function is a bunch or description of the scattering angles, and the mean cosine of those angles, that's exactly the G factor. And the G factor, in case of minus 1, it means that all photons scattered back. G factor 0 means it's isotropic. It may be scattered in any direction. And and in case of G factor 1, as you can see, it's predominantly or only forward scattered. In biological tissue, G factor is around between 0.7 and close to 1. And that's how the phase function of scattering looks like. And that's the function what they use for approximation through the G factor of the phase function. That's basically it. The scattering coefficient times 1 minus G is called a reduced scattering coefficient, and that's the inverse value of, characterizes exactly the transport mean free path, what I was talking about, when after this distance, the photons become totally diffused, okay? So just kind of like to put it all together. What's interesting and what is kind of like non-linear about that? Because of the scattering, we have a phenomenon in biological tissue of a certain trapping of the photon. If we send the light inside the skin, so it may form so-called subsurface peak. It means that the fluence actually under the skin, when light enters, becomes higher than the incident light. That's about it. So this is the intensity, the fluence rate of the incident light here. And as you can see, inside the tissue, it becomes higher. Why? because of the scattering, it produces some sort of like a trapping effect for the photon and keeps basically the fluence rate high. And this process, when we have a multiple scattering and optical trapping effect, by the way, it's described by the so-called the effective damping coefficient, which is mu effective, and this is the formula for that. And we also characterize the value, what's called the effective mean free path length, which is 1 over mu a, but instead of the mean free path what we were discussing before, instead of mu s, it operates with mu s prime. So why it's important? Because the effective mean free path basically provides us with effective penetration depth, meaning that we can provide a visibility of a certain object in the scattering media up to these depths. After that, the contrast, or more exactly, the signal to noise ratio deteriorated, and we cannot obtain a reasonable or registerable signal to collect the data. Okay, now like in terms of the quality, so far we were talking about like metrics of the scattering. In terms of the quality, we have two types of scattering, and that's prescribed by X factor, which is 2pr over lambda, and r is radius of the object. And if x is much less than 1, we're dealing with release scattering. It means very small objects produce a release scattering. And release scattering is proportional to the force power of the wavelength. So that's why the blue light is scattered much higher than red light. When X factor is about 1, so we deal with misscattering. And misscattering depends on the wavelengths as a first power, which means it's more or less flat. And when the X factor is, or we deal with big objects, so then we can use actually geometrical optics approximation. So that's how it looks like. With release scattering, it's pretty much isotropic in all directions. With misscattering, it's predominantly forward scattered, and the larger the particle, the more forward the scattering is. And that's an illustration. So we deal with, or we observe release scattering, a blue light, because blue light is scattered much higher. That's a release scattering because of the air particles, which are air molecules, which are really, really small. And misscattering, that flat, because the dependence on wavelengths at the first order. And because of that, we have a mixture of all scattered lights, which would give you a white color, and that's what about cloud. And the scattering is provided air droplets or water droplets, which is much bigger and comparable with the wavelengths. In biological tissue, nevertheless, we have both release scattering and misscattering. So actually, I'm talking about scattering so much because it's one of the kind of like probably pillars of the difficulty providing the biological imaging, the scattering. And in the shorter wavelengths, we deal mostly with the release scattering because of the small structures, usually like intracellular. And from 500 and longer wavelengths, we deal mostly with misscattering, and somewhere like in between 400 and 500, we have a mixture of both release scattering and misscattering, and that's basically again prescribed by the size of the objects. Okay, so this is the last probably from the introduction. That's from a single low-order scattering to multiple scattering. If we consider that, with a single or low-order scattering, we can see a good contrast because basically it's not smeared, it's not blurred by the scattering. But once it becomes a higher order of scattering or multiple scattering, a lot of scattering events, it's becoming less and less distinct, and that's what happened and what's illustrated here, right? So from ballistic propagation, when we can obtain a very good contrast to and including the low scattering, and this is the transport mean free pass, and that's the relationship. Once we got more than one transport mean free pass, so the photons become totally diffused. So how we would describe what the formalism behind that? So far I explained the phenomenology, but now I would like to talk about the maths. So first of all, of course, the first tool what we have, it's a Maxwell equation. In terms of biological tissue, it's actually quite difficult to solve because you have a lot of unknowns. There is a transfer theory, which is approximation of the Maxwell equations. And it actually considers light penetration and light distribution as a diffusion, like any diffusion process. And that includes actually a few components. I probably will mention that later on. The third approach is a diffusion theory. At the distance more than transport mean free pass, when all photons become multiple scattered and diffused, so we can apply a spherical symmetry. And as a result, we end up with a diffusion theory, which is an approximation of the transport theory. So this is a sequence of kind of like simplifications. And this is just an example of Maxwell equation solution on a single particle, on the ensemble of particles, and basically that's a prediction of the light distribution at a certain parameters. So coming back to the transfer theory, because this is probably the most I would say accurate description of the light distribution in biological tissue. So it comprises of a few terms. So the result is the change in the radiance in the certain direction. So and that equals to the negative term of the attenuation. So that's why it's negative. And it also includes the gain because of the scattering, meaning that certain photons are coming back and contributes in the radiance. So remember these peaks, the subsurface peak of the fluence rate, that's exactly because of the gain due to the scattering of this optical trapping effect. And the equation operates with the coefficient we just discussed: absorption coefficient, scattering coefficient, extinction coefficient, or transport coefficient, or attenuation coefficient, which is the superposition of absorption and scattering coefficients, and the phase function. And the phase function is approximated by the G factor through Heine-Greenstein function. And that's probably the last to mention. The analytical solution for diffusion approximation can be present here, and that's including the reduced transport scattering coefficient, and once again 1 over mu s prime. That's when the photons become diffuse and multiple scattered. Okay, and that's the main kind of like mathematical tool we operated. So the idea behind this ballistic photon imaging, what we developed, is that once we restrict the numerical aperture, we would restrict basically the angle of the scattering, and we started collecting from, let's say, a generic image collection, the photon collection, to gated photon collection. So instead of like collecting all photons, we collect only photons that would be less scattered, because the gating would be provided by the lower NA. And we started to simulate based on the transport equation. We started to simulate that by Monte Carlo simulation, considering only scattering in the first phase. As you can see, we could reproduce the subsurface peaks because of the Chapin effect, and changing the numerical aperture, we could change basically the location of this peak, and that's the parameter what we use. It was, by the way, quite well supported by the earlier publication, when we were checking kind of like, okay, so how about our results, how well it correlates with results before. And as you can see, with a reduction of the numerical aperture, the contribution of the low scattered photons actually is growing here, or vice versa, with increasing the collection angle, the contribution of the low scattered photons is actually dropping, and the contribution of multiple scattered photons is growing. Okay, so then we added absorbing contrast, and that's what we can see. Again, that was our simulation to check whether applying the restriction in numerical aperture, we can improve the contrast. As you can see, the numerical aperture 0.5 will lose the contrast quite early when we add kind of like an additional layer of scatters. And when we restrict the numerical aperture, our imaging or absorption contrast remaining much crispier, and the contrast is much better. Yeah. So that was kind of like we published this work, and that was very theoretical at that moment. Okay, so then we started to build the device based on this principle. So we took the chip of CMOS center, we removed the infrared filter, because that's what create an unnecessary gating. And we actually glued the fiber optic plate with a lower NA, and then we glued this fiber optic plate right on the chip. And the second modification of this device was that instead of like placing the restrictor of the numerical aperture, the fiber optic plate with a low NA, right on the chip, we made that distance located, and then we placed a readout of the objective on the surface of the fiber optic plate on the proximal side, while the distal would be collecting the data. The light sources we arranged around the fiber optic plate, and that's an example of the image what we started to obtain. And we tried to figure out like which schematics would work better, because that was could be found only experimentally. So those are examples of fiber optic plates with restricted NA. And this is the schematics of the experiment. What we did, we applied, let's say, the USF 1951 USF target at the bottom, and then we poured intralipid of a certain concentration, and we changed this concentration, mimicking basically the scattering properties of the skin. And we placed the fiber optic plate either close to the subject, and we were reading that out, or we glued the fiber optic plate directly on the chip. And we were trying to figure out, okay, so what works better? So this is our schematics for the second approach. When we place the fiber optic plate inside the Petri dish with intralipid, and we were reading that by the microscope objective, and then collected by the camera. We also applied 3 different wavelength ranges, and those wavelength ranges we were trying to figure out, okay, so which light, which color would work better for our purposes. Okay. And that's a result. Okay, so with a Microsoft objective, so we were trying to compare, let's say, without gator, without numerical aperture restriction, and with, and where the contrast, and comparing the contrast. Okay, with a microscope objective, so there is no numerical aperture restriction. That's the depth of the intralipid inside the Petri dish. As you can see, we can observe the contrast only up to 400 microns here. While applying the what we call a spatial angle filter to facilitate the ballistic photon imaging, the contrast was visible up to 900 microns or even more. So this is the continuation. So that's from 0, right? 900, 1900, and up to 2600, which is basically 2.6 millimeters. As you can see, the advantage of ballistic photon imaging was immediately proved basically with the first experiments. So the improvement comparing with, let's say, just a bare objective, was from 400, we started to provide a good contrast much deeper, up to 26 or even 2700. So the improvement was practically 8-fold. And this is shown just as a plot. And then we started to work with different concentration of intralipid. The higher the concentration of intralipid, basically intralipid, that's some sort of like a milk, which applies in ICU, and that's for the feeding the patients in case they cannot swallow, they cannot like eat normally. So that's some mean basically to deliver certain nutritions in the human body to make them survive. And interestingly, this type of kind of like milk, it's very close in terms of the scattering properties to human skin. And that's an example of improvement for different concentration, as you can see, applying the numerical aperture restriction, we can improve and still provide a decent contrast up to 500 microns, even at the high concentration of intralipid. So what is closer to human skin, which is 2%, that's basically what's providing the contrast up to 100. So that's what we were comparing between the numerical aperture. We were applying fiber optic plates with different numerical apertures, and that's shown on the plot. And that was comparing the numerical aperture 0.17 versus 0.55. 0.55, that's pretty much the standard fiber optic plate, and 0.17, that was the customized fiber optic plate. And if you compare, you see that the lower NA fiber optic plate provides us the contrast much deeper if you compare that with 0.55. So 0.55 stops facilitating the contrast. That's approximately 1.2 millimeters. And the 0.17 gives us a decent contrast. Decent, I mean like more than 10%. That's what the human eye can distinguish. And up to 3 millimeters, which was kind of like a threefold improvement. Excuse me. Actually, I was told that the presentation is about 60 minutes. So I was counting on 60 minutes. So that's why I'm kind of like a little bit confused at the moment. I can interrupt my presentation, that's for sure, but yeah, okay, good. Thank you. All right, so that's presented through the plot. And also what we did, we were trying now to compare the different wavelengths. And what we found that the green actually range works much better, which is sort of like understandable because in green range we have a Q-band of many absorbers, more exactly hemoglobin. But also, even for the scattering, even just for interleaved only, for some reason, the green range also was working the best. And that's a presentation of these results on the plot. So that was the imaging, and that's on the plot. And that's, again, it's a Michelson contrast versus the interleaved signals. Okay, so once we kind of like experimentally, we build the devices, we experimentally proved that it really works, and actually it worked even better than we expected. And then we started to build some sort of like phantoms and mimicking the capillary structure, the vessel, the blood vessel structures. So that's our microfluidic phantoms right here. And those are the imaging of the real phantoms we created. And we started to try applying those with absorbent channels, filling the channels with hemoglobin, and pouring on top interleaved. As a result, it would create a pretty close phantom, a pretty close structure to a human skin. The channels with hemoglobin would mimic basically the blood vessels, and the interleaved would mimic the scattering properties of the skin. So, those are, again, the results we obtained. And that's a conventional image, all right? And those are the image, what we obtained with a third modification of our device, which was, we applied just a single fiber, and that single fiber was scanning. And the single fiber, again, was with the numerical aperture reduced. And this approach would definitely let us, or would enforce us, to collect data longer than with the imaging setup. But at the same time, it also gave us an opportunity to change the pixel size, the pitch, and we had a wider variation of the numerical apertures. Okay, those are basically all three modifications. So we had the objective plus camera plus broad white light, and that's what we're comparing with. And then the second was the chip, the spatial angle filter placed on the chip. And the third modification was the fiber optic plate plus objective plus camera. And we were reading out the proximal end of the fiber optic plate by the objective of the microscope. So as a result, that's what we obtained. We obtained actually an image of blood capillaries which were totally hidden under the skin. We could not see them, but nevertheless, applying the ballistic photon imaging and the element what we call a spatial angle filter, which is a low NA fiber optic plate, we started to obtain actually nice images of the capillary loops. And just to explain a little bit the anatomy, that we have the capillary loops actually quite easily visible at the nail fall, which can be obtained and imaged even without our technique. But on the phalanges at the same time, or rest of the body, the capillary loops are much deeper and we cannot see them. But nevertheless, as a result, with the help of our technique, we started actually to see not only the capillaries, but we also can develop the resolution of this image to see, as you can kind of like probably distinguish, the elements of blood, more exactly red blood cells and its aggregates, that's flowing through the capillaries, which actually opens a lot of like potential applications. And those are examples of what we collected throughout the body on phalanges, nail fold, just to compare, and it was enough to collect the data and obtain basically the image of the capillaries. Yeah, and we were trying again different techniques, applying, and eventually we ended up that the fiber optic plate, which is about to be read out of the Microsoft objective, these schematics seem to be working the best with the highest resolution and the deepest interrogation depths. So then we started to describe that in terms of trying to retrieve certain processing of the image and coming out with certain parameters what we can retrieve from this image, like width of the limb of the capillary, distance, periodicity, or height of the capillary loop, and so forth, and even providing eventually the statistical analysis or histogram of distribution of those parameters. And that was a preparation basically for us to start collecting numerical or quantitative statistics, which can be later on probably used for machine learning and AI processing. Okay, so in the end, so this is the conclusion. The ballistic photon imaging, what I developed with my students and employees in the lab, improves the sampling depth by 3-5-folds versus conventional or objective-based imaging, maintaining similar absorption-based contrast. So filtering the diffuse photons by the NA restriction leads to a drop of the signal. This is the price we pay. Once we gate the highly scattered photons, obviously we start to obtain much less photons, up to maybe 20 times less. So this price what we pay should basically warn that in application, or let's say we apply more light, and as a result we may lead to overheating of the sampling area of the skin. Actually, we did not bump, or we did not like really face this effect so far. So it was still enough to maintain the illumination level within the reasonable level. But I probably should mention that because of the gating, we need more light to provide. And in terms of delivery of the project, like within last 6 years, we had 1 PhD defended, 3 master students defended. We obtained 3 patents and published 5 peer-reviewed publications we're still working on. And, and the full patents umbrella successfully filed. and provide us an opportunity to proceed with commercialization of the technology. So the last slide is the way how we think to create kind of like a commercially feasible device. So we start with the chip, the fiber optic plate glued to that, then fiber optic conduit to provide the illumination, the cover, and as a result, we obtain some sort of like a scanner. And with this scanner, we can basically visualize and analyze the pattern of the capillaries of the surface. And this pattern actually changes dramatically in case of a number of diseases. First of all, cancer. In case of the cancer, the nice periodic pattern changed to scrambled pattern, and that can be easily seen by such a device. So all we need, basically, it can be connected through Bluetooth or Wi-Fi with a detector, with a display, and all we need to provide just some sort of like light coupling media between the distal end of the device and the surface of the body for better or reduce the light losses. Okay, last but not least, that's my acknowledgement. First of all, of course, to my students and co-workers, my collaborator, Dr. Gennady Saiko, Dr. Vlad Toranov, the PhD student who defended her thesis based on developing this technology, my current students, Mina Bagderi and Kiana Daliri and Fiona Zerai, that's master students, and my postdoc, Aditya Pandya, who already left the lab and located in California. And this is my group, about like 17 people altogether. And the funding, of course, from Natural Science and Engineering Research Council of Canada, Canadian Cancer Society, and my hospital I'm affiliated with as a scientist, and yeah, support from my colleagues overall of the department. And in the end, I would like to thank you for your attention. And if you have any questions, so please.
Okay, so anyone have question about the lecture? Okay.
Yes, just a slightly naive question. Right? I wondered what the obstructions would be to providing a slightly wider field of view with the technology.
Yeah, that's an excellent question. So far, we developed already approximately 1 inch, which gives us kind of like plenty of space. At the same time, we found the optimal size is probably 1/2 inch diameter, because it gives us an opportunity to provide a reasonable illumination, more or less homogeneous. So that's where we are. And with a 1/2 inch, actually, you can make a scanning of a certain human body part in terms of diagnostics pretty fast.
Okay, okay, thank you.
So maybe let's thank again to Professor Douplik's good talk.